So wonderful to be here and so happy that this is actually happening.
Well, we're all very happy that it's happening because it wasn't sure at all that we could make anything happen after these restrictions that came in 2 days ago. But we really, really wanted to make it happen. We want to not skip a year. We'd like to honor these wonderful scientists and artists, and we're going to do it. We're going to do it.
I think that is really, really important, and I really laud everybody for trying to make this happen. And I'm very happy that we arrived at the Royal Dutch Academy of Science. Let's see if they let us in.
Yes, let's see.
Hello, hi, this is Mr. Carvalho and Thomas Erdbrink for the ceremony.
Thank you.
Thank you.
Thank you.
The Heineken Prizes. Almost 6 decades now. Where does that passion for science, humanities and art come from? Well, it started with Freddie Heineken's curiosity. His inquiring mind, always looking for new, meaningful solutions, was what connected him with scientists, scholars and artists. And the recognition with a prize was his way of making sure they received the respect they deserved for their important work in society. The awards opened doors to publicity, to continuity, and new ways to success. The Royal Netherlands Academy of Arts and Sciences selects the winners for the prizes, and that they choose well is evidenced by the fact that they picked 14 later Nobel Prize winners. And for the future laureates, the Heineken Young Scientists Awards were launched to inspire a new generation and to motivate them in their quest to breakthroughs. Just as the prizes were called into life, what more will their curiosity bring us?
The Heineken Prizes over the past 5 decades have come to be the most prestigious international science award in the Netherlands, while also honoring the arts. We are very pleased to have you all here with us for this unique live edition of the award ceremony. My name is Thomas Erdbrink and I am so honored to be your host today. I'm a journalist for the New York Times and a documentary filmmaker. But a very big welcome to you, our viewers. And of course, a very big welcome for these 2 important ladies here in the room. Welcome, Charlene de Carvalho, chairman of the Heineken Foundation. And also a big welcome to Ineke Sluiter, the president of the Royal Netherlands Academy of Arts and Sciences. Naturally, we would have loved to see everybody in person here with us this year. Charlene de Carvalho, please tell us all why we should really be watching this live event.
Well, like Thomas, like you say, it's really sad we can't have our normal celebration. It's usually a very great event for me anyway, and I think it's much appreciated by the laureates as well. However, we didn't want to completely ignore the fact that these wonderful people have had these prizes, and I'm very, very grateful also to Ineke and to the Academy for allowing us in these circumstances nowadays to still continue and to do something at least and have a happy moment and a congratulating moment for these incredible scientists.
Absolutely, it's really, really wonderful to be here. Ineke, I can imagine these are challenging but also yet very interesting time for science.
Absolutely. I mean, if the pandemic has told us anything, it is that when there is a crisis, people really know where to turn for answers. And that was science and scholarship. So, and not just virology and epidemiology, but also, what does this do to our way of living together, for equality, the economy, health in general, well-being, education. So yeah, these are important times for science.
It's truly all-encompassing what is happening in the world at this moment. And Mr. Carvalho, when we sort of focus down to what we are seeing today, what really fascinates you about the laureates?
Well, the laureates are obviously exceptional people, and they are. I am very honored and humbled meeting these people. Sadly, I'm not going to meet them for more than a very short period on screen, and I hope that I will in the future have a chance to get to know them a little better. They have devoted their lives and their incredible intellect to improving our lives and to improving the world around us, and that is something we should celebrate.
Yeah, absolutely. I think it's really, really wonderful that we are making the best of this difficult situation. And just like often scientists who are persisting in their field, we are also trying to continue. And just, Ineke, before we move on with the program, I'd like to ask you, why now especially is it important to invest in science?
Well, I actually think it's almost always important to invest in science, because investing in science and scholarship is investing in our future and our well-being and our young people. And investing through prizes like these mean inspiring people, rewarding achievements that have already happened, and then inspiring a whole new generation to find the solutions for the
Wonderful.
next.
Wonderful. Thank you both very much. And please allow me to explain what we will be doing tonight, as it is a bit different from what you're used from us normally. For the first time, the Heineken Prizes has decided to build the ceremony around themes that unite the laureates and their work. Working interdisciplinary is more and more the norm in most fields. And we are looking forward to connecting the laureates and their fields of research. Today, we'll give some insight in what drives these extraordinary people, how they connect and how they impact our daily lives. The coming hour, you will meet our laureates. They are 9 outstanding scientists and 1 engaging artist. In pairs, they will discuss 5 themes where they share a common interest in their field. And let's start with our first theme, the organ that defines us as humans. Well, what else can it be than our brain? First, before we start with that, let's get some background on our laureates from some key members of the jury of the prize. And you are the chair of the jury of the Heineken Young Scientist Award 2012. But let's just start right away. What makes Anna van Duivenvoorde such a promising scientist, and why has your jury awarded her with the Young Scientist Award?
Yeah, our committee was really impressed about her innovative research on brain development, and in particular the topic that she has, studying teenagers, the development of brain in teenagers, social behavior, how they take risks, saying yes or no to risks is especially important also today, so it's a very timely topic. And also what impressed us is that Anna is really an interdisciplinary scientist. She works together with psychologists, neurosciences, but also statistics. And moreover, she's a real example to young scientists, very inspiring personality.
Yeah.
So she really deserves this.
I have had the fortune of already meeting her, and she's truly a wonderful and very interesting person. And before we get to her, I would like to introduce the first laureate of the Heineken Prizes, Carl Dasserot.
He's by far the most creative neuroscientist of the last decade. With his methodology, by which you can stimulate individual cells with light, you can be much more sophisticated. You can stimulate specific networks and uncover the specific functions to that network. When you know that, you can also use this for treatment. So his technology is now being used for trials to treat patients, psychiatric patients, neurological patients, patients with blindness, etc. The nice thing is that the tools that he developed are so robust, anybody can use it, even you. Together, the whole neuroscience community at large is now uncovering how the brain works, mainly thanks to the tools that he developed. And on top of that, he's a really nice person. So he discovers many different things, and he's willing to spread it all over the world. So there's hardly any neuroscience lab that's not using his tools.
Welcome, Jacques Neefjes. Truly talking about a very, very interesting laureate. Please do tell us more about him.
Yeah, Karl Dijsselhout is a special person. I think he should have had this award actually for 5 or 6 perhaps disciplines, and let me shortly explain it. So he gets it for medicine, and that's because the brain has a lot of effects, of course, on many diseases, and he studies these. So the diseases like depression and like schizophrenia. The question basically that he wants to address is how does it work? So where does it come from? And he uses technology for it. He developed technology that allows to basically use light in the brain to set up particular reactions that you would associate with normal social behavior, like eating behavior or like trying to meet other people. So he can basically put that on and off with light. So you could say, well, it could also have been cognitive research. And he did also develop basically technologies that are so beautiful that he could also have had the award for art. So in a way, and what I think is that he also, if you think about it, the fact that neurons in our brain can control our normal social behavior is in a sense also a form of philosophy. So what are we? Are we our brain? And are we our brain cells? Well, these are issues that he is studying using extraordinarily beautiful technologies.
Yeah. I was told that he actually found a way to sort of switch on and off certain feelings and emotions. Is that correct?
Yeah, that's correct. So he uses actually a protein from algae that he introduced in the brain of a mouse. And that algae protein is sensing light. And then it basically puts on and gives a sort of a very small current. And then the brain thinks, something is happening, I have to do something. And then he changed that algae protein to do the exact opposite. So basically put signals off. Yeah. So he can, with light, different colors of light, he can basically just control behavior.
It's almost unbelievable. Jacques, I really want to thank you. Carl, we'll be seeing more of you later. Thank you again. I say, let's meet these laureates in person and let's discuss how they view the brain. The brain is a shared field of research for Karl Dijsselhout and Anna van Duivenvoorde. Karl focuses much of his research on understanding brain circuits, for instance, brain circuits that are involved in depression. And Anna's quest is to learn more about the link between brain development and teenagers. In this first Heineken Prize conversation of the hour, they will talk about how brain development might change in these COVID times. And of course, this is caused by video calls and quarantine. And they'll also discuss if this is different for teenagers versus adults. Carl, I think you were joining us from Stanford. Is that correct?
I'm here in Palo Alto, the town where Stanford University resides. So thank you very much for the tremendous honor. And I'm looking forward to this conversation.
Absolutely, Carl, and thank you for being here with us. Anna, how are you feeling? Everything okay?
I'm okay.
Yeah?
Thanks,
okay.
Thomas.
So, as this is all about talking together in the short time that we have, Anna, I would ask you to just kick it off right away. What's your question for Carl?
Hi, Carl. I'm so honored to talk to you. And, well, you did groundbreaking work on optogenetics, so the technique to switch on and off neurons with the use of light. And I was hoping that you could share your insights on what your research on brain circuits and behavior have learned us about sensitivities in the developing brain.
This is a really great question. It's very important, particularly today. We know that the pandemic has caused tremendous stress to young people. In some way, it's a form of extreme stress. And we know that that can have lasting consequences. I'm also a psychiatrist, so I think about this a lot from the clinical perspective as well as the basic science perspective. I know, as you do, that early life stress and early life trauma can have lasting consequences. We don't know exactly how that works, but we're getting clues, ideas that come from the science. And I'll give you one interesting example. There is a form of altered brain function called dissociation, where people who are subject to trauma will very often separate their sense of self from their body. And this sounds like an almost science fiction-like concept, but it's amazing how consistent the description of this state is from people who've experienced trauma, emotional or physical. And using optogenetics, just in the last year, we've been able to make headway on understanding how such a thing can happen, how the sense of self can be dissociated from the body. We've been able to play in certain patterns of activity, provide them, writing in instructions, if you will, into a particular part of the brain that we saw was abnormally active in a mouse that had been given a dissociative medication, a medication that can cause dissociation. We had seen an interesting altered pattern of activity in the brain, but we didn't know if it was causing the behavior. Optogenetics gave us that key leverage, that causal understanding. We could then play in, provide exactly that pattern of activity that we had seen the dissociative drug had elicited. And amazingly, we found that we could elicit directly, using light, using optogenetics, targeting a very specific circuit in the brain, we could cause a dissociative-like state in the mice. And that in turn let us go to a human study where we found a very similar phenomenon in the same conserved ancestral region of the brain, and we saw a dissociative state in a human being as well. So this is an example of how the basic science of optogenetics can give us insight into these very deep, mysterious questions that for a long time have been thought to be outside the reach of science.
Carl, I think that is truly almost incomprehensible for an outsider, but the effects will be so wide-lasting and it will have so many globally results. But do you also have a question for Anna?
I do, and this is something that came from my clinical experience as well. So I've had to start to give my psychiatric appointments virtually, of course. And this is something that was always possible, but was never really allowed by the psychiatry establishment because it's, at one level, it seems as though you can't communicate enough social information over video. But one thing I noticed is that younger people seem much more comfortable than older people, and they are able to communicate much richer social information over a video call than older folks. And so this is an incidental observation, but it's also really important. If we understood this, we could help people much more effectively. And so I wonder if this is something that you have insight into, if you could help us understand this or make it work better for the older people.
It's such a timely question. And well, we know that the teen brain is very wired to social information. They are very sensitive to social acceptance, social belonging, and this is not necessarily a bad thing. This also makes them quick social learners in a way. And well, I think if we think about whether this learning or whether these relations are different between online and offline communication channels, I think there's a lot we don't know yet, and therefore it's also an empirical question that you're asking. And we know that the real world is much more complex, it's very rich, and making social connections also helps in our well-being. As a psychiatrist, you must know that, but also if we looked at studies in our teens, being away from school, missing friends, having more negative thoughts and mood, that was all reported also in our teens. And in a way, I think these online connections might actually help deepening the physical relations that we already have, or the real-life relations that we already have. And well, teens may be even more attuned to that, or even more accustomed to that. And in a way, well, I think so the combination of both would be just very beneficial. And I know we're maybe running out of time, so unfortunately, I would love to ask you more advice for young researchers, and I hope we will get a chance to meet again and chat more about that.
Absolutely. Thank you both so much, and we all would wish for this conversation to last much longer. As we are live and you are watching from home, we will move on to our next laureates. Now more than ever, we communicate on so many levels, but how do we really speak to one another? The next theme is our communication. Yeah, I'm not really stalling here. This is actually what the work of the next laureate is about. Carl, welcome back. Why is Mark Dingemans' research so absolutely, totally intriguing?
And unconventional, because I would like to say that, because very unconventional. We're impressed about that. He wants to study why languages are as they are. Like, for instance, the word, huh? Eh? It got worldwide attention. And that's actually the same in every language. It has the same meaning. And he's studying these patterns and also working together with neuroscientists and psychologists. So he has a very good communication skill himself, I would say. And also, he's a guy who really likes open science. So he wants to distribute it all over the world to disseminate it. And that really impressed us a lot in the jury.
What I found really impressive that he spent months and months in Ghana examining this. Truly remarkable. Thank you, Carl. There will be more from Mark soon, but allow me to introduce the second laureate of the Heineken Prizes, Robert Zatorre.
First of all, he tackles an area that is outside of the scope of mainstream cognitive science and cognitive neuroscience, namely music and speech. But at the same time also why we appreciate music so very much, by its relation to structures that are the reward systems in the brain that produce dopamine if we find something rewarding. And he has shown that there's a strong connection between, on the one hand, these areas that analyze music, and at the same time the reward systems that make it pleasurable.
I think the most inspiring aspect of his research is that it covers not only the scientific field, but also touches upon artistic matters and artistic questions.
He has been showing that musical training makes your speech understanding also improving by the coupling of the auditory and motor cortices. So that could be potentially something which could be helpful in one form or another for persons that have a certain problem in hard hearing or understanding speech and so on.
Welcome, Peter Hagort.
Thank you.
Thank you. I hear that Robert he's also trained as an organist.
Yes, he started out as an organist and has a musical training himself before he went off into the field of science.
Do you think that really influenced his work?
I'm sure that, of course, because one of his domains is music, so in that sense he knows it not only from studying it from the outside, but also from the inside, so to speak. So he is a gifted musician himself.
And what was it that really struck the jury in Mr. Zatorre's work?
Well, I mean, one of the things that we share with other species is, for instance, the fact that we have visual perception, that we have memory. But there are two things where we stand out in the animal kingdom. And these are speech and music. That's something that are uniquely human skills. And they're based on, of course, the acoustic patterns that reach our ears. And Robert has been able to make foundational discoveries about how the auditory cortices in the human brain extract the relevant information, such as pitch and the temporal modulations that are both crucial for speech and music. And moreover, and that's another important component of his work, he has been able to show why some of us appreciate music, by a technique called positron emission tomography, where he showed that if you listen to music, the fact that rewarding is based on the fact that there's a connection to the dopamine system in the brain that gives off dopamine as a reward signal.
Basically sets off something in your brain.
But then also there are people that don't appreciate music, so they're called so-called anhedonia. And that's because of the fact that this connection between the auditory cortex and the reward system is not functioning in the same way as for those of us who appreciate music. So this explains mechanistically why some of us have strong emotional feelings with music and others don't. And I think that's a fascinating discovery, because it not only tells how these sound systems work and how they're extracted, the information extracted in the brain, but also how they're connected to other networks that in one way or another make it important or less important.
Yeah, so if you're a music lover, it's in the end, it's all in the brain.
In the end of the day, of course, the music has to come from the outside, but how it's been handled and how it's appreciated,
it happens in here. Thank you, Peter. Thank you, Carl. I say it's time to meet Mark and Robert and see how they discuss and how they communicate. Sounds are universal and they come in different forms. For instance, your favorite pop song, but also noises like or Robert Sartore and Mark Dingemans are experts when it comes to these sounds, both in their own fields of music and in the field of speech. For the next Heineken Prize conversation, we will get more insight and see if this use of sounds that we use for our social interaction are also used when singing. Welcome, Robert. You're joining us from Montreal, Canada. Can you hear us? Hello, very good to have you. Just a very short question, Robert. What is the best song you were listening to today?
Oh, well, I think it would have to be some excerpts from the Marriage of Figaro by Mozart.
Well, this is, of course, a very excellent and classical choice. Mark, how many times have you been counting my mms?
Well, you're at 8 now, and that's perfectly fine.
I should not be that worried?
No.
Okay, well, thank you. That's really great. I think enough from me. Mark, you have a question for Robert.
Indeed. Robert, it's such a pleasure to talk to you. Part of your work focuses on why we find music pleasant. And in language, we have something similar, of course. And there, what we find pleasant and what we accept as beautiful is often also shaped by context, of course. You know, a weird sentence can sound better due to context. Now, in music, I've noticed something similar in listening to, Grammy Award-winning people like Esperanza Spalding or Jake Collier. They use stacked chords, even microtonal modulations, but you don't even notice it because they build up the context so well. So what's the role of context in shaping our expectations and our feelings of pleasance?
Yeah, thank you for that question, Mark. That's an excellent question, and I think there are obviously very, very strong of context which are very, very important in music. A given chord or a given tone, even whether it will complete a phrase or whether it will elicit surprise, depends entirely on the context in which it is presented. And we can actually look at that in a formal way. For example, in some of our research, we've looked at the structure of a melody in terms of its overall predictability, which you can measure in terms of entropy, for example. And so if you have a highly predictable structure, then people actually prefer to have some unexpected surprises thrown in there. Whereas if you have a relatively chaotic structure, hard to follow, because it's a high entropy, and then under those conditions, people actually prefer to have some way of being able to anticipate what's coming and have that expectation fulfilled.
Mm-hmm.
So what we find is that the reward system is very much sensitive to exactly that balance between predictability and surprise.
Paul, do you also have a question for Mark?
Yes, I was very intrigued by your very original work, because I think linguists are always looking at the parts of speech as in, words and verbs and things like that. But you're interested in these other sounds, and to my musician's mind, it strikes me that these are kind of the percussive elements of talking. And so I just wonder whether you think that they're completely integrated with the rest of the more classical linguistic system, or are they really something separate? And in particular, are they handled via the same neural circuits that handle all the rest of speech, syntax and semantics and all of that, or are they a parallel system?
That's a wonderful question. Thank you. I think the answer is going to be a bit of both, but they are clearly different from many other words. And the reason is they actually have sort of a meta-linguistic function. So they really operate on a different level of function. And that's why, first of all, we don't notice them, even if we use them so much. And it's second of all, why they sound a little bit different from other words. So I think you may be right that also the neural circuits that we employ to use these words might be slightly different, although to an important extent they will also overlap, I'm sure, with language. Because, I mean, in a way, in fact, we wouldn't have language as we know it without these small words that streamline our everyday conversations.
Mark, is there also something something personal maybe that you want to ask Mr. Sartore?
Well, so I'm really interested in the fact that you have a background as an organist and as a scientist. And I'm wondering, are you still practicing as an organist? And how does that still now influence your work, perhaps?
Yes, well, I'm fortunate to be able to maintain some music on the side. And in fact, in my laboratory, a really beautiful digital organ that we use to study the sensory motor interference, because to play the organ in particular, you need to coordinate all four of your limbs because you play the pedals with your feet. So that gives me an opportunity, both to do some fun science and also to sneak in there when none of my students are using the organ and play a little Bach. So the best of both worlds.
I think you two should definitely have a coffee together and play the organ a little bit. Thank you, Robert Sartore. Thank you, Mark Dingemans.
Thank you.
And before we go on to our next theme, Our Truth, we will look at the provoking work by the 2018 Heineken Prize for Art winner, visual artist Erik van Lieshout. Artist is free and artist makes art. Thank you, Mr. van Lieshout. For the coming theme, which is very much under siege, we have decided to pair two Heineken Prizes laureates. Both are committed to finding answers to one of the oldest questions in our history. What is truth? Therefore, our next theme is Our Truth.
Lorraine Daston has asked questions and is still asking questions about, say, the ways, means and reasons of our knowledge that no one has asked like this before.
She shows that all the things that we usually think are timeless have a history and have developed over time.
She is really a fundamental collaborator, someone who works always with others and yet inspires by her very shining example.
She's not a leader in the traditional sense. She doesn't exert any kind of power. And having this non-hierarchical approach where people collaborate on the basis of equality, that makes her a great leader. She simply loves to do so. She doesn't see it as work. It's a kind of oxygen to her.
What makes her inspiring for young scientists is first and foremost that she is willing and eager to work with young scientists and to inspire them to write better and more interesting history.
Welcome, Josine Blok. I can see you smiling. You're really very, very pleased with this laureate, right?
I'm so happy. I mean, she is really one of the most eminent historians today, and I am absolutely delighted that she is our winner. She so much deserves it. Her work is so rich food for thought for everyone, not just historians, but far beyond that. So I'm very, very happy.
Can you give us an example of what really strikes you in her work? What really makes her tick according to you?
Well, indeed, she asks questions, as I said in this very short video, that no one has asked before. And this is, she investigates how we work as scientists and scholars and how this has come about historically. And some of these issues seem so self-evident. For instance, the notion of objectivity. We all take it for granted that scientists and scholars are objective. I mean, that is one of the standards of our work. But this has always been like this before. I mean, this has a history. And she has investigated with a colleague how in the early modern period the ideas of objectivity were developed, both the ways and means in which scientists brought this idea about by creating measures, weights, methods, making pictures or atlases or whatever kind of methods they had developed to create this kind of objectivity in knowledge. But she also investigates how this kind of work creates a certain kind of person, that as a scientist and a scholar, you also adopt a certain attitude. So what she calls the scientific self. So the people that we are, we have come about through this process. The way we work nowadays has a very deep history, and she has shown that.
A thought-provoking scientist.
Absolutely.
One of our great laureates.
Yes.
Please allow me to introduce our next laureate, Ansuya Blom.
Personally, I'm really very touched by the ways in which Ansuya manages to give expression to inner worlds that are normally always enclosed and we never have access to. And she really is not somebody who is telling others how the world should be. On the contrary, this would really be very counter her work and her ideas. She always, in her own work, also looks for the brittle, the things that are uncertain, and in that uncertainty and brittleness, she finds beauty. And I think this is also what she allows in young artists who are often searching and finding their own ways. And if you can, see that there is beauty in this uncertainty, I think that will help artists and, young people to find their own place and their own voice.
Welcome, Patricia Pisters. You are a jury member for the Heineken Prize for Art. And unfortunately, Frits van Oostrom, the chair of the Art Prize jury, isn't able to join us today. But thank you very much for being here with us. And I can see you smile very much also. You speak of Mrs. Ansuya's work as being brittle. I understand you both, you became familiar with her work while working in an archive. Please tell us what happened.
Yes, I was working in the archive of the Dutch Film Museum and encountered her work, and I was immediately touched by the ways in which she portrays inner worlds, but also the worlds and perspectives of people that we usually don't know. They are forgotten in history. They are, they disappear in the folds of history. They are not taken up in the canon. So, for instance, psychiatric patients. One of her works, she makes a lot of films, besides also other work. One of her works is called Lola Magneta, and this is giving us the letters and the thoughts of a psychiatric patient that we know from the reports of her psychiatrist, but her own perspective has never actually been heard. And these are the kind of fragile realities and worlds and inner worlds, hidden worlds that Ansuya's work gives us, brings to us, and brings into the light.
It's extremely interesting that she chooses to highlight those we do not see that often, and that this comes across in her work so very, very clearly. Thank you, Patricia. Thank you, Josine. And I think it is time to meet these two extraordinary women in person and hear their views on the truth. The question, what is truth, is a shared quest for all of us. And of course, as being a journalist, I can especially relate to this, especially in these times. And truth is also a common interest in both Lorraine Daston's and Ansuya Blom's work. Although one has a scientific approach and the other has the point of view of a visual artist, both explore the room there is for more than one truth. So how can we trust knowledge or scientific knowledge when it's constantly changing? We might find out during our next Heineken Prize conversation. Lorraine, these are special times. I understand you are in Berlin, Germany.
Yes.
Thank you very much for being here. Thank you. Ansuya, also, thank you very much for being here. Lorraine, I understand that you will be asking the first question for Ansuya. Please go ahead.
With great pleasure. Ansuya, I hope that we will have the pleasure to meet in person sometime.
Yes.
And also, I hope to have the pleasure of seeing your art. There are truths of art, just as there are truths of science. And your own work has set a very powerful example by crystallizing emotions for which we almost do not have words. And my question for you is there a particular emotion to which you come back over and over again in your art, trying to crystallize it, to make it visible for all of us?
Thank you for that question. And I really look forward to meeting you as well. I mean, to answer your question, I would say it's not an idea, it is the fact of anxiety. I think living in the times that we live in now, of course, it's a time that provokes anxiety enormously. It is an anxious time, it's a real time. But anxiety has been maybe at the root of, or the basis of my work for many, many years, for different reasons. And what I find compelling about this time is actually that makes me think back to previous times that I have experienced. Equally, I'm also aware that anxiety we feel right now is in a way comes on in a space of luxury, in the sense that the West hasn't been exposed recently to big traumas, but I'm acutely aware that at the moment, in different parts of the world, people who also already live in very fraught conditions now get Corona on top of it. And so their situation is very, very complicated. And I also think that's not without reason that today, or these days, Black Lives Matter comes to the fore, the situation in nature, global warming. And I had to think back of my first experience with the pandemic, some call it epidemic, in New York, when in the early 80s, when I had a friend there, I still have him as a friend, and AIDS came about. And what he and thereby me, but so many others experienced was something so unknown and so fraught and so with a true sense of danger. But besides the fact of that particular anxiety, it also made for a general sense of nervousness, unknownness, and I see it in a funny sort of way as a Kierkegaard. I should switch. Kierkegaard talks about anxiety. He's a philosopher I'm interested in, and he says nothingness constitutes anxiety. And perhaps these days we all feel this nothingness because we can't see a future. We don't know where this is going to end.
Yeah, you really capture how we all feel. And I'm wondering, Ansuya, what is your question for Lorraine?
Lorraine, yes. So I much enjoyed your book about nature, against nature, and so you have the scientific approach to go back also and to look at how truth can come about in relation to knowledge. But I was wondering, are there certain times that make for certain questions, or are certain times forbidding certain questions?
It's a wonderful question, and it highlights something about how partial our knowledge is. That is, if we were to imagine our knowledge as a map of the world, it would be one of those medieval maps in which there are big, dark spaces populated by monsters because we have not yet had the curiosity to probe in that direction. And one of the real questions, central questions for the history of science is, why does our curiosity flow into some channels rather than others? Sometimes it's a question of urgent need. So at the moment, virology and epidemiology are our pole star sciences. But at other times, it is pure curiosity. Why should we want to know what the stars are made of or how insect sensory systems work? Our attention is very individualized, which is why it's so important to have a diversity of knowers. Each of us brings a different pattern of attention, a different pattern of curiosity, and if we pursue inquiry collectively, we have a better chance of panoramic knowledge.
I really think that we should make a monthly TV or online program with these conversations, because I just want to hear more from the both of you. It's so interesting. But at the same time, I also can't wait to hear from our next laureates. One truth we are confronted with every day, of course, is our changing climate. So it seems very fitting to move to our next theme, our nature. Welcome back, Carl. It's good to see you again. And we'll be talking about the work from one young, outstanding scientist, Freddy Rabau.
Yeah.
Please tell me more about him and why did your jury pick him?
Yeah. Freddy is a special person. He's by far the youngest of all.
What's his age?
I think he's from 1988, so he's 32. Very, very young still. We're impressed about what he did. He's a photonics person, nanophotonics, and he looks at very tiny nanocrystals of materials. And the interesting thing is, and the discovery that he made got worldwide attention, also in the press. He showed that when he couples light or electricity in that nanocrystal, that depending on the dimensions of these crystals, the light changes in wavelength, and you can even see this by your naked eye. And this got immediate attention, not only from the scientists, because this is basic science, but also from the people in the, for example, the solar industry and the display industry. So it may be that one day his discovery ends up in what we buy somewhere in our instruments.
Freddie
So,
makes our new television.
really, really impressive.
Well,
And,
we'll get to meet him soon. And this gives me also the moment now to introduce our next Heineken Prizes laureate, Corrina Leclerc. And let's watch.
She's really at the forefront of studying the interaction between climate change and the global carbon cycle. And she does that in a very interdisciplinary way. Her research has been very influential both on the scientific communities but also at the policy level. Because climate change has been debated, you know, whether it's true that what humans do actually influences the atmospheric CO2 level. But her data are so explicit, and she was very convincing to the politicians and the general public, and that is very necessary. She actually narrows the variation in the data so that we can make better predictions on what's coming up. Well, I think every scientist with such a great achievement is inspiring by itself. But I think overall it's her interdisciplinarity that I think is very inspiring to show students and colleagues to look, you know, further than their own little discipline. And that's so necessary for this field of research.
Welcome, Louise Vert. Such a diverse laureate. What do you really admire in her work?
Yeah, I admire a lot in her work. And one thing I already mentioned is that interdisciplinarity that you need for such a complex research field. Think about it. We produce, as humans, we produce all these greenhouse gases, and we burn fossil fuels and wood and all kinds, and it comes in the air. And then we have to realize that how does nature deal with this? You know, part of it is absorbed by vegetation, and about 23% is absorbed by the oceans. And so the oceans, they actually, you know, help us to not change the climate immediately so fast. It could go slower because of this absorption. But how does that work? What processes are there? And you cannot just study that with a single discipline. So she has to deal with the biogeochemistry. She's a physicist, but she also dug into the biological system, because it's this biological system in the oceans that also play a crucial role in absorbing this CO2, of course. On the other hand, she showed, for example, that's really in the Southern Ocean, that it may actually go down with that possibility of absorbing more and more CO2 in the ocean. So it's actually, you know, with warming up. Yeah. So, and if that happens, then there is a positive feedback, of course, and then there's even more global warming. So you can see the complexity of that. She's a leader, and with her whole group on all these different aspects. And I really appreciate her also as a person.
Why?
And you should look at her TEDx Warwick recently, I think, or last year. She gave that. Yeah, and so it's very humoristic as well. And she actually shows how she deals as a scientist with this complex science field. And she says, I'm not going to persuade any more politicians or anything, but I have the data, and very good data. Data on modeling and observation together that make it possible for us to predict what this will be in the future.
I'm definitely going to look at that, and I urge everyone to do the same. Thank you, Louise. Thank you, Carl. And if our nature is really what keeps us alive, then how can we find a better balance? Let's see what Corrina and Freddie have to say about this. Nature can help society a step further towards a more sustainable planet. but we are a consumer society. How can we find a balance between sustainability on the one hand and quality of life on the other? Our laureates, Corrina Leclerc and Freddy Rabau, work in a different field, but are both interested in this fragile balance. I'm sure we will find out more during the Heineken Prize conversation that is themed Our Nature. Corrine, I understand you are in Norwich. Is it cold over there?
Yes, in Norwich in the UK.
In Norwich in the UK, of all places. And Freddy, did you, when you started studying chemistry, actually not that long ago, did you ever expect to receive this much praise at such an early age?
Thank you, Thomas. No, I never set out to for this, but it's a great experience, absolutely.
Fantastic. It's such an honor to have you here both. Freddy, you have a question for Lorraine.
Yes.
Go ahead.
Professor Leclerc, I see that you're an excellent scientist, but also heavily involved in discussions with policymakers and the public. My question to you is, how do you balance these two tasks, and how is this balance, maybe against your immediate will, shifted over the years?
Thanks for the question. I'm actually incredibly lucky that there is interest for my research outside of the research area. I find it really interesting, in fact, to have the opportunity to share my knowledge. And you see with the COVID crisis now how important it is that scientists go out and speak about their science at times of crisis. I think that moment for me has always been very enriching. I would not do it all the time, though, because my oxygen is really in the science. And so for me, that always takes me back home in my research environment.
Corrina, I understand you also have a question for Freddy.
Yeah, absolutely. Freddy, I mean, incredible work that you do with those nanocrystals. I understand there are some implications for, or potential, I should say, even for solar panels, which is one of the possibilities for deploying renewable energy and tackling climate change. But I hear from your research that you could make them even more performing now. And can you tell us a little bit how and where you see the limits would be for that performance?
Yeah, solar energy will of course be very important in our future society. And you may not realize that solar panels that we have on our roofs are not so efficient yet at all. Like 20% is a very good solar panel. And the main reason why they are not so good yet is because the solar spectrum contains all colors of light, and the solar panel is usually only good at converting one particular color. So the sunlight contains red, orange, yellow, green, blue, etc., but also invisible colors like infrared and UV. So this whole spectrum is not converted so efficiently. So that's the main factor that limits the solar harvesting right now. And these nanocrystals that can absorb light and emit different colors, they can be very useful for that, because we could, for example, use them, and that's one of the things that I'm also working on. We could use them to convert the sunlight before it enters the solar cell, so that it has the more preferable colors before it enters the solar cell. And that way, we could boost the efficiency of the solar cell significantly.
Great.
Yeah.
You have another question for Corrina?
Yeah,
Maybe
so
a personal question?
Solar energy will be very important, but our society may also need to capture carbon from the atmosphere to suppress the greenhouse effect. My question to you is, what could the role of the oceans be in there? Would it, for example, be useful to extract carbonate from the oceans and then let this equilibrium do the rest, and in that way reduce the concentrations in the atmosphere?
Yeah, thank you. I mean, you're completely right that we most certainly will need to absorb carbon out of the atmosphere. And of course, we have the tree who can do this for us on land. In the ocean, we could actually artificially pump carbon in the ocean, either through biologicals, so enhancing the biological processes, but that would have pretty negative impacts. We can do it through engineering also, just pumping it in the deep ocean. And in fact, if we did this, we would short circuit 1,000 years of ocean circulation, because at the end of the day, when we end the emissions of carbon dioxide, then the ocean will absorb most of the CO2. However, engineering-wise, it's not that easy to capture the carbon, to pump it. It will take a lot of time to design these technology, and it cannot be done instead of reducing our emissions, but it could really be done just for the remnants at the end that we can't really bring down to zero. So I'm afraid that your technology with solar panel is much more likely to help us in the immediate term than the ocean engineering.
Let's hope you will both help us, because of course everyone is hoping that we can find a new balance between our own lives and with nature. Our next and final laureates, I believe and I hope, are ready for us. They will have a conversation about the elements that we are all made out of. Let's go on to our next and final theme, our cells. Carl, last laureate of the evening for you, Meta Rustenberg. Meta has been it's been part of a team that has been working on finding a vaccine for malaria. Please tell us why you have picked her.
Yeah, Meta is a special person. She is really a pioneer in human disease models and infectious disease models. That's her topic. She's the first worldwide who used a genetically modified malaria vaccine. And she actually vaccinated normal persons, healthy persons. And she could demonstrate that this was effective. That's the first time this is done, especially in this COVID period. That's also very important because, of course, we also would like to see what happens if we infect normal persons with COVID. How dangerous is it? So she is consulted a lot at the moment. She's in the newspapers a lot. And she is very important also in the WHO, where she actually is one of the advisors of the malaria team there. So we are really impressed. And the last I would like to say, she's a very open personality, very approachable, and a great mentor for young scientists.
I've met her and she's indeed truly fantastic. Which brings me to another truly fantastic person, our next and final laureate for the Heineken Prizes, Mr. Bruce Stillman.
Especially of him is that he started very early on a topic that he continued throughout his career. And at a very young age, he saw the importance of the results he got, but also the experiments he did. Amazing. He found a complex of proteins, an origin of replication complex, that is the start of the replication of DNA. And then with ATP, it goes to the next step, and it continues to do so, and at a certain moment, DNA is replicated. But this is the origin of that idea. And that is what you have for the, obviously, for the reproduction of everything. Viruses, tumors, human beings. Therefore, you have to replicate the DNA before the cells divide into two. And he found the protein-DNA complexes in a unique way. For young people, I think it's a message that if you believe in what you do, even if other people say, do something else, continue on what you do. The more people say you shouldn't continue, you should continue.
Welcome, Bert Meijer. Before we get to you, I've been told that Mr. Stillman is working in a lab called Cold Spring Harbor Laboratory, and that this lab is actually an incubator for Nobel Prizes. And I've also been told that you are all watching over there, so a big hello to you from us in Amsterdam. What do you think, Bert? Is his research a potential candidate as well?
That's a very difficult question, but what he worked on is just spectacular. I think Bruce worked on a question that everyone is interested in, and how DNA is replicated. And when he started, he said nothing is known at the time of the late 70s about the cellular DNA synthesis. So I thought, let's just start to try to find it out. And that's what he did. He purified many proteins that are needed for the replication, but also once a single strand of DNA is made, that it is a protein that kept it and protected it, as well as the histones that everyone knows where the DNA is all wrapped around. So fabulous work, very important, even as today. Because as we know, if the DNA synthesis goes well, we get a nice replicate. But when it doesn't go well, we go into the disease. Or we're interested on how cancer goes. And that was one of his prime interests, not only to find out how the synthesis is, but also what you can learn for diseases and especially cancer. I think that was also the reason why he went to the United States from Australia, to be very close to figuring out how you could solve that immense problem. And so I think he's an excellent candidate and winner of this most important and oldest Heineken Prize in Biochemistry and Biophysics, due to the fact that he also gives a lot of credit to the people he works with. Really an amazing person that really deserves this beautiful award.
Thank you, Bert. It's a beautiful word from Mr. Stillman. Thank you, Carl, for the whole evening introducing all these fantastic laureates. And now we will find out what connects Meta and Bruce, and we will hear their views on our cells. DNA replication proteins are fundamental to our bodies and are often used as targets for novel drugs. In this final Heineken Prize conversation, we welcome Bruce Stillman, known for his discovery of various important proteins. And we also welcome Meta Rustenberg, who works on a candidate malaria vaccine. And both work on a microscopic level, but that microscopic level has a huge impact on our personal lives, on what we are made of, on our cells. Bruce, I hear you have a passion for yeast. Is that correct?
Yes, actually, I study yeast and human cells. And it's actually quite appropriate, given that this is the Heineken Prize, because this yeast that I study is actually used to make beer, bread, and wine. So it's quite a nice connection there.
Indeed, indeed. And Meta, I'm so happy you are here, because beyond all the wonderful things you do, you are also active in battling COVID-19, and you had to undergo actually a Corona test today. And what was the outcome?
I was tested negative, otherwise I wouldn't have been here. But it's better safe than sorry, right?
Absolutely, absolutely. Meta, you have a question for Bruce. Please go ahead.
Thank you. It's an absolute honor to meet you today and to be able to ask you a question. I was wondering, gene regulation is so complex and it involves many proteins, and the function of these proteins is very often redundant, so other proteins can take over, and proteins function in complexes. And it worries me because very often these proteins are used as targets for novel drugs. And as a clinician, I'm worried whether we can actually grasp the full complexity of this network and really predict what a novel drug will do in vivo. Are we able to actually do that?
Well, it's a good question. In fact, it's one of the things that drove me to study DNA replication, because as we know, cancer cells are constantly proliferating and abnormally proliferating. And my idea was that if we understand DNA replication, we may be able to understand targets for cancer therapy. And one of the things that happens in cancer cells is that they break down the regulation of this process. And so when we understand the complete replication process, one can devise drugs against certain proteins that in cancer cells will work more effectively than in normal cells, because we have normal cells in our body that are proliferating. And this, and there are now new drugs coming out of the studies that the field has done. For instance, some of the proteins we've worked on, there are others who have developed small molecule inhibitors of them. But it is very complex, and redundancy is the reason why biology works.
Bruce, thank you. Do you also have a question for Meta?
Well, first of all, congratulations, Meta. I've read about your work, and it's quite amazing. And I also want to say it's amazing that you're a physician and scientist that are doing both. And I think that's an incredible achievement. Genetic variation, of course, comes from DNA replication. And one of the things I worry about in developing therapeutics, and particularly vaccines, is the genetic variation. How much variation in the work that you've been able to do to develop attenuated parasites, how much is genetic variation and species variation going to affect the ability to treat malaria?
Yeah, genetic variation indeed is the agony of vaccinologists. Basically, finding something that doesn't change, that's conserved, is the holy grail. Because what we try to do is beat evolution. And the malaria parasite, like any other microbe, I suppose, is continuously changing, trying to evolve and avoid our immune system. So particularly these targets that we think are suitable for vaccine candidates are very diverse. And change very quickly, which is obviously a problem when developing vaccines. And the same goes for attenuated parasites. If you want to, take out a protein, you have to make sure that it's not redundant and it's really essential to the parasite. So yes, you're right. I think DNA replication does underlie a lot of processes in medicine. Which basically brings me to my next question, and it's a bit of a personal one. You've had such an impressive career working on DNA replication since 1979, and I was wondering, how did you come to work in this field? Was it something that you chose to do, or was there some kind of serendipity that led you there?
Well, thanks for the question. It was a bit of both, actually. I wanted to do medicine since I was 11. And when I got to university, as you often do, I worked in a lab. And I got very interested in research. And I switched and started pursuing research, and because I wanted to work on cancer. But the serendipity comes in because I went to a seminar in the medical school and heard about a particular area which ultimately I worked on, and decided that's what I want to solve in my career. And so it was kind of a bit deliberate as well. And so I came to Corrina eventually after doing my doctorate in Australia, not intending to stay here for a long time. I've been here for a long time. And it's a great place to work.
I must say, I really, really, really love hearing you two talk. And I can see, Bruce, that you really love the laboratory where you're working. Please stay there and keep everybody happy. Keep on doing what you do. Thank you, Meta, also for being here. And we have heard them explain their promising research. We know what they have achieved. We have listened to their conversations with the Heineken Prizes laureates. And now, please join us for the official presentation of the 4 Heineken Young Scientist Awards 2020. And of course, normally we would have had flowers and hugs and kisses, but we all know what times we're living in. In other years, Charlene de Carvalho's son Alexander and daughter Louisa would hand over the awards. Today, we're doing it a little bit different. We're going live to Louisa. Louisa, welcome. You're joining us from London. You're missed.
Yes, hello. Thank you for having me. I hope you can hear me well.
Absolutely, we can hear you very, very well, and we also hope to see you here in the future. And we had to change many things, but I understand that you're also honoring the laureates with a new symbol this year. Is that correct?
Yes, absolutely. And let me first say it's a complete honor to be able to virtually present these prizes, which I understand are sashes created by the artist Carl Martin. And it is such an honor to be able to hear about all the amazing work these scientists have done and are continuing to do, and especially to be able to represent the young scientists on behalf of my family and continue the legacy of my grandfather. And every 2 years we have more participants, and it's just such a shame not to be there in person with you all. But next year we'll be able to all get together and meet properly.
Absolutely, absolutely. And if I understand correctly, we will now see. And now, because we're lucky enough to have Meta here also right now, we will see her being presented with the sash in the room next door. And there she is. Very, very happy, very, very happy. Thanks to all these great winners. Many congratulations to the laureates of the Heineken Young Scientist Awards. Really fantastic work, and I really look up to all of you. Now, the youth has the future. But of course, the day is also about honoring those amazing scientists and an artist who are changing, improving, and inspiring our lives right now. So now, the ceremony for the Heineken Prizes. But as you can understand, and to our great sadness, we can't all be present in one room today to honor all these magnificent achievements. We're scattered around the globe, but I hope today a little bit together in spirit. And again this year, in this challenging year, has once again shown us the importance of science and creativity. And while we are unable to hug and shake hands and congratulations, we are handing you over the awards today just in a little bit different way. I would like to invite Charlene de Carvalho and Ineke Sluiter to, in a symbolic gesture, present the prizes. After each turn, there will be a brief word by the laureates. And to you, our laureates in Norwich, Berlin, Montreal, and beyond, we kindly ask you to keep your prize ready because we already shipped it to you. Let's start off with our friend from Cold Spring Harbor, Bruce Stillman. Charlene de Carvalho, may I invite you?
Thank you, Thomas. Bruce Stillman receives the Dr. H.P. Heineken Prize for Biochemistry and Biophysics 2020. Congratulations.
Thank you very much. Here's the award, and it's wonderful. I want to thank the Academy and the committee who selected me for this very prestigious award. The award that is a microscope that was invented by Antoine van Leeuwenhoek, and he actually saw yeast in 1680, and this is yeast that I study and have a yeast in beer. So I think it's a very good connection to him. We've studied things in much more detail than he did. And I want to thank my collaborators and students and postdocs who contributed to this, and also to Cold Spring Laboratory for the amazing support that I've had over the years. But finally, I want to thank the Royal Netherlands Academy of Arts and Sciences for recognizing these awards, but in particular for this award, recognizing fundamental and biology research. This is really important to drive science forward. Thank you very much.
Thank you, Bruce. Mr. Carvalho.
Carl Deiseroth receives the Dr. A.H. Heineken Prize for Medicine 2020. Congratulations to you.
Thank you very much. It's a tremendous honor to receive this recognition. It's a particular joy to receive it, and this is a silver lining of the pandemic, as we can do things like this with our family, as well as connect with our colleagues and friends. So I'm deeply grateful to my family, my wife and children, who've done so much to help me along this path. Also deeply grateful to all the collaborators around the world, in Europe, United States, and elsewhere, and all the students and postdoctoral fellows. This recognition of basic science is very timely. Science represents some of our highest aspirations, and we all are looking forward to working together in the future to help the community as a whole. Thank you very much.
Thank you, Carl, and so great to see your family. A big hello from all of us to all of them over there in Stanford, in Palo Alto. Ineke Sluiter.
Lorraine Daston receives the Dr. A.H. Heineken Prize for Historical Sciences 2020. Congratulations.
You might have some screenshots.
Thank you very much. I'd like to thank the Heineken Foundation for its generosity and its public spiritedness in supporting the arts and sciences. The Royal Netherlands Academy of the Arts and Sciences, which has been a beacon of the Republic of Letters for over 200 years, to be esteemed by the scholars and scientists and artists of that organization is an honor indeed. And finally, to everyone who produced this ceremony, thank you for your gallantry and good humor under very difficult circumstances and making it a true celebration. Thank you all.
Lorraine, thank you. You are a total star. Thank you very much. Ms. Sluiter.
Corrine Leclerc receives the Dr. A.H. Heineken Prize for Environmental Studies 2020. Congratulations.
Thank you very much to the Heineken Foundation and the Royal Academy. This means a lot to me. I thank also my colleagues of the Global Carbon Project and my incredibly talented research group, the University of East Anglia, Peter Liss for his precious mentorship, the Royal Society, NARC, and the European Commission for their support over many years. And finally, my family, my mother, Nicole, my daughter, Marianne, and my husband, Paolo, for their unconditional love and support, which means I'm a human at the end of the day. Thank you.
Corrine, thank you. You are a human at the end of the day, but really one that helps us strive forward. Thank you. Mr. Carvalho.
Robert Zatorre receives the CL de Carvalho Heineken Prize for Cognitive Science 2020. Congratulations.
Thank you so very much. Here is the beautiful prize, and I'm glad to see that there's a brain inside there. I very much want to thank Madame de Carvalho and the Heineken Foundation and the Academy for this amazing effort that supports both science and art. I think it's really important to recognize scholarship and achievement in both of these domains that I think are really essential for humanity's survival. I really want to thank all of my many brilliant students and research fellows. They're the ones who've had all the innovative ideas and have really contributed the most to our success. And of course, I want to thank all of my colleagues, both here in Montreal and many other places. And finally, I want to thank my family for their love and support, and especially my closest collaborator, my wife, Virginia Penton.
Robert, thank you very much. And we are very much hoping to see you here in Amsterdam in the future. And maybe you can come to our Grote Kerk and play the organ once. Thank you. Mr. Carvalho.
Finally, someone who is actually here, Ansuya Blom, receives the Dr. H. Heineken Prize for Art 2020. Congratulations.
Thank you. KNW, Heineken to begin with, actually. KNW. Also the jury and those who nominated me for this award. Honored. I also want to thank, I want to thank all those who worked with me on my films over the years. And in accepting this, or accepting, but in getting this award, I'm also thanking people that have been very influential in my life, that have been Vivian Bourgogne and Stuart Morgan. And my biggest gratitude, in all honesty, goes to my husband, Mark Lynn, for helping me with my work. We've been collaborating since day one, and for being who he is and with who I have the great fortune to share this precious life. Thank you all very much. I'm honored.
Thank you.
Asuya, thank you very much. We had a little bit trouble understanding the first part of your speech, but you were saying how grateful you are for all those who cooperated with her films. And we are very happy that you are here with us. And as it turned out today, we are neighbors. So this is a very funny coincidence in this magnificent prize. And I just want to say that here we have now all the laureates of the Heineken Prizes, 5 on the screen and 1 in the room here. I say, let's do what we can do. Let's give them all a very big hand. Yeah! Congratulations, you can keep up your prize. We are very, very happy in Norwich, in Palo Alto, in Montreal, in Berlin, and of course, in the laboratory. Thank you very, very much. And of course, I want to thank you, Miss Charlene de Carvalho, Louisa in London. Thank you to the Heineken Foundation. And we must, of course, thank Ineke Sluiter. Your organization, the Royal Netherlands Academy of Arts and Sciences, has been a great host for our ceremony here. And we have been so lucky to be here in your beautifully renovated building right here in Amsterdam. And all of us here at the Heineken Awards, we want to thank you, our dear viewers, because it's your continuing interest and support that keeps these very important Heineken Prizes going. We are so grateful for having had you here with us today. That's it for me, Thomas Erdbrink. And for the next edition, let's just say that here at the Heineken Prizes, we trust in science and we trust in your creativity. And we trust that you will make sure that we can all meet again soon in person. See you in 2022.